All right, everyone, welcome. Thank you so much for joining in on this presentation. I hope everyone is keeping safe during this COVID pandemic. Um, Today I'm going to be talking about hidden hearing loss and the effects of synaptopathy. And this is a relatively new topic in the field of otology. The goal of my presentation is to give a broad overview of what this is and to talk about some potential therapies in the future to treat it. As a warning, I'll be going over a number of basic science papers. My focus will be to keep things as simple as possible and to not overwhelm you with the details. Okay. I have no disclosures here. So I'll start off by talking about the traditional dogma of hearing loss, specifically to that which relates to acoustic trauma. Then I'll talk about what hidden hearing loss is and what it means to have synaptopathy. Then I'll the three main effects of synaptopathy, talk about some of the issues with contemporary audiometry and the way we measure hearing loss. Then I'll conclude by talking about some theories as to how synaptopathy can cause tinnitus. And then I'll talk about some therapies in the future, some exciting therapies that can treat synaptopathy. So to start with some background, auditory transmission, as you know, begins as sound vibrates the tympanic membrane and the middle ear ossicles. The energy from this then vibrates the perilymph in the scala vestibuli and the scala tympani. In this process, the basilar membrane is vibrated. And as you know, the basilar membrane has one row of inner hair cells and 3 rows of outer hair cells. The inner hair cell, the function of the inner hair cell is to transmit auditory information to the brain via the spiral ganglion neurons. At the base of a hair cell, there are these unique proteins called presynaptic ribbons, and their function is to tether neurotransmitter-filled vesicles. And the purpose of this is to allow for rapid neurotransmission as glutamate is released to the postsynaptic cleft and allows current to enter into this nerve and allow for information to proceed to the central nervous system. Now, I know that this awfully looks like something that we've seen in the news recently. It looks like COVID-19, but I assure you, there's no coronavirus in the inner hair cell. If any of you know of anything like this, then let me know. I'd love to write it up. The human cochlea has approximately 3,000 inner hair cells, and this is really not a lot of receptor cells, because if you compare this to the olfactory cell system, there's actually 50 million olfactory receptor cells. And if you compare that to the visual system, that's 100 million rods and cones in the retina. So given just the number of the fewer number of receptor cells in the inner ear, it really highlights and underscores the importance of these structures and their connections, as the whole auditory system and our ability to hear depends on these few structures. So the inner hair cell is connected to multiple spiral ganglion neurons. The spiral ganglion neuron, by nature of its name, is a ganglion cell, and its purpose is to connect the peripheral auditory system to the central auditory system. And the ganglion cell is a bipolar cell. It has one nerve ending that brings afferent information from the inner hair cell, and another nerve that sends information to the cochlear nucleus. Studies have shown that on average, there's approximately 15 or so different spiral ganglion neurons that connect to the inner hair cell. And this is what you're looking at is synapses per inner hair cell as a function of position in the cochlea. And you're looking at data from mice. So that means 15 different of these cells are connecting to one inner hair cell. And remember, there's no branching here. It goes from one synapse to one nerve to one spiral ganglion neuron. So I'll be using those terms interchangeably during my talk. And if you look at the differences in animals, you see that the synapses per inner hair cell and the cat, at least in this study, had the highest number of synapses per hair cell. And if you look at the humans here, there's actually fewer number in humans than compared to other animals. So when the inner hair cell depolarizes in response to sound, this opens up voltage-gated calcium channels and allows for a neurotransmitter, namely glutamate, to activate these nerve endings. There's two different types of synapses and nerves in the cochlea, and they are the low spontaneous rate nerve fibers and the high spontaneous rate nerve fibers. And as you can guess from the name, the low spontaneous rate nerves spike a fewer number of times compared to high spontaneous rate nerves in the absence of a stimulus, which is really interesting. High spontaneous rate nerves sometimes can spike up to 100 spikes per second in the absence of a stimulus. So these nerves are actually segregated anatomically with respect to where they attach to the inner hair cell. The low SR nerves attached to the medullary side of the inner hair cell, and the high SR nerves attached to the pillar side of the inner hair cell. And besides their anatomical differences in the way they attach to the hair cell, there's also three main functional differences between these nerves. So the first difference is that if you look at this graph, threshold as a function of frequency, the high SR fibers have a lower threshold for activation. This peak is lower than the low SR fiber. What this means is that that these nerves are more readily activated with responsive sound. And if you look at the width of this curve, the high SR fiber has a broader width compared to the low SR fiber. So this means that they have a broader range of activation, a broader frequency range of activation. The last difference is that the high SR fibers have a shorter, smaller dynamic range before they saturate the response. So here you're looking at the discharge rate as a function of sound intensity, and you see that there's a much narrower window that the high SR fiber goes up in its activation before it saturates. And also, if you look at the low SR fiber, the slope here is much smaller, and it covers a broader range of sound intensity. In terms of actual numbers, there's 60% that make up the high SR nerves, and 40% make up the low SR nerves. And taken together, this is a lot of information, but basically it comes down to the high SR nerves are thought to be needed for detecting sound. It has a much lower threshold for activation. So you can think of this as having a higher sensitivity to sound. And the low SR fibers are needed for hearing and noise because they're more active above the threshold. So it's suprathreshold sound detection. So you could think of this as allowing for higher specificity of sound. So now that I've explained the organization of the inner hair cell and the spiral ganglion neurons, I'd like to go into what people used to think happens when the cochlea is exposed to loud noises. So until recently, it was thought that after acoustic trauma, the most vulnerable element in the cochlea was the hair cell. It was thought that the hair cell was the primary target for acoustic trauma, and that the synapses and the spiral ganglion neurons degenerated as a consequence of the hair cell death. So in this study published in the year 2000, they actually exposed chinchillas to loud noises, 86 dB, for 2 days. And then they looked at their basilar membrane to see what would happen to the cells in the basilar membrane. So you're seeing a photomicrograph here of 1 row of inner hair cells and 3 rows of outer hair cells. And you clearly see that in the middle, there's this large chunk of area that's missing cells. And these authors tried to quantify this, and they certainly found that there was inner hair cell and outer hair cell loss in this basilar membrane. And concomitantly, they found that there was threshold shifts in audiometric studies, and this correlated with the amount of sound that these animals were exposed to. So the idea was that there is a primary event, which was cell loss, hair cell loss, and spiral ganglion neurons. They noticed that they degenerated at a different time course, so they thought this was a secondary event. So to summarize the traditional dogma, sound causes hair cell death, and this causes ribbon synapses and nerves and spiral ganglion neurons to degenerate. Now, before I go into why now this theory or this dogma is in question, I have to take you back to a study, older study published in 1955. And this is an important study because it tried to understand the relationship between the auditory thresholds and spiral ganglion neuron loss. So in this study, they performed a craniotomy in cats, and they selectively cut parts of the auditory nerve in order to remove or kill off certain populations of spiral ganglion neurons while maintaining the hair cell population intact. So this is the main results of their study. And what it's showing here, each data group here represents an individual cat. And what you're looking at is ABR thresholds in the top row. You're looking at hair cell numbers, hair cell counts in the second row, and spiral ganglion loss in the last row. And for this, the more black you see, there's more spiral ganglion loss. And what this is showing is that you can actually maintain your auditory thresholds, your audiometric thresholds, as long as you keep your hair cell numbers intact. Because it doesn't seem to matter whether there's ganglion loss in the middle of the cochlea. In this cat, there were ganglion loss kind of all throughout. It really takes up until 80 to 90% of spiral ganglion loss until you start seeing a change in the ABR, in the hearing test. So what this study showed was that audiometric thresholds do not sufficiently capture spiral ganglion loss. So what happens when your thresholds recover? So this study tried to understand what happens to the synapses and the spiral ganglion neurons when you actually recover your hearing thresholds. And in this study, they exposed mice to 100 dBs of sound. And to give you an idea of what that sounds like, that's like being in a loud concert. It's an approximation of that loudness. And they exposed mice to 2 hours of that. And then they looked at what happens to the thresholds. And this is a well-documented phenomenon. It's called the temporary threshold shift, in which initially you have a hearing loss. And over time, in this case, over the next 8 weeks, you get a recovery of these thresholds. So this is a DPOAEs, these are ABRs and compound action potentials. And many of you listening in probably experienced this temporary threshold before. Whenever you went to a loud concert, you would be exposed to loud noises, and initially you feel like your hearing is diminished, but you notice that over the next couple hours to couple days, that your hearing comes back. And what you're experiencing there is actually a temporary threshold shift, and it's thought to be mediated by outer hair cell function. But the authors here wanted to understand what's happening at the synaptic level when you get this phenomenon. So what you're seeing here is a control of mice not exposed to noise. And you see these little red dots here represent synapses. And this is an outline of an inner hair cell. And you see a robust number of synapses in the control. One day after noise exposure, you see a dramatic drop off of these synapses. And what's interesting is even 2 weeks after exposure, what you're looking at here are spiral ganglion neurons. You start seeing some changes, some loss of spiral ganglion neurons. But more than a year after the exposure, you get a dramatic loss of these cells. And if you look here, this is showing you that the nerves that go to the outer hair cells, the efferent nerve fibers, also get affected. There's a number here that gets reduced here. So what this study showed was that even long after noise exposure, you can get changes in spiral ganglion counts, and that synapses are really affected during loud noise exposure. So that's pretty convincing evidence that there is synaptopathy present in animals. But what about synaptopathy in humans? Because this is experimentally very challenging to do, to prove the presence of synaptopathy in humans while they're alive, obviously, They try to look at cadaveric temporal bones to see if there's any evidence of that. And in this study, they harvested temporal bones in adults post-mortem, 40 hours after death. They harvested the temporal bones, they fixed them, dissected out the cochleas, and stained them for neurofilament. And what you're seeing here is one individual, and this is the apical region of a cochlea, or the basilar membrane, and you see these green streaks. They're representing neural fibers, and this is the same cochlea, same person, but in the base. And you see that there's a dramatic difference between the two regions. And this is the osseous. These are actually the spiral ganglion neurons here. These are the inner hair cells and the outer hair cells here. And this is showing you a cross-section of this yellow dotted line shown here. These authors then went ahead and counted the number of hair cells in these individuals. And I think there were 9 individuals, but I'm just showing you a representative here. And if you look at the black dots, this is counting the inner hair cells. And you notice that overall, the inner hair cell counts are preserved throughout these individuals, maybe except for the basal region. But this isn't the case when you actually look at the type 1 fibers in the per inner hair cell and ribbons per inner hair cell here. There seems to be a dramatic difference as you age. So for example, if you look at this 89-year-old gentleman, he had a low number of type 1 fibers per inner hair cell, as well as ribbons, but had and this is the same person here, had intact inner hair cells. So what this is demonstrating is that, number one, that the synaptic elements and the neurons are more vulnerable than the inner hair cells. And number two, this seems to be correlated with age. So the next question becomes, is synaptopathy an early indication for auditory functional decline? And this question was addressed with this study, and they used a mouse model of age-related hearing loss to see what happens, what's the time course of cell loss among the inner hair cells, spiral ganglion neurons, and outer hair cells. And what you're seeing here is that the inner hair cells throughout age actually are relatively preserved, and outer hair cells, as expected, actually decline with age. What's striking is that if you look at this gray curve here, this is the number of spiral ganglion neurons, and the x-axis now is age. These steadily decline, as do the ribbons, the synaptic ribbons, but the inner hair cells, which is shown here in gray, actually are preserved relatively throughout the age of the animal. So this goes to show that that the synapses and the spiral ganglion neurons may be the more vulnerable elements in the cochlea, not only with acoustic trauma, but also with age. So what is hidden hearing loss? Hidden hearing loss is intimately related to synaptopathy. And the idea here is that initially you have a normal hair cell and its synapses. Once you have some level of noise exposure, you start losing these synapses. And then eventually the nerve fibers themselves degenerate. Because you have an intact an intact hair cell, you preserve your normal thresholds. However, because you have synaptic loss, you're not able to really hear accurately in noise. So hidden hearing loss is hearing loss in the setting of a normal audiogram, and the hidden part means that it's hidden under normal thresholds. And the premise here is that hair cells are not the most vulnerable elements. It's actually the synapses that are the most vulnerable. So to summarize, hidden hearing loss, some noise leads to ribbon synapse damage, and sometimes you can have excess noise that leads to even hair cell damage, but it's not, the synaptic damage is not a consequence of the hair cell death. So what's the mechanism behind synaptopathy? As I've explained before, the inner hair cell, when it gets activated, When it gets depolarized, it releases neurotransmitters into the postsynaptic cleft of these nerve fibers, and glutamate activates NMDA and AMPA receptors, which allow current to enter into the nerves. Now, studies have shown that if you infuse the cochlea with AMPA, which is a glutamate agonist, you can get these morphological changes in the synapses, and it causes them to swell like this. And in fact, if you actually just expose a guinea pig to noise of 130 dBs for 15 minutes, and you look at their basilar membranes, you actually see these synaptic swellings as well. And the morphology is quite strikingly similar to just adding glutamate into the cochlea. So this is thought to be mediated by glutamate excitotoxicity. And if these authors did an interesting experiment, they exposed guinea pigs to noise, 130 dBs for 15 minutes. In one cohort, they gave artificial perilymph, and in another cohort, they infused riluzole, which is thought to inhibit release of glutamate. So if you look at, there's a striking difference between the two. You have these nerves, the same synaptic nerve swelling with just artificial perilymph, whereas you don't really get that much in the riluzole group. And these authors also measured compound action potentials, and they found that riluzole was actually otoprotective compared to artificial perilymph. So this all goes to show that glutamate excitotoxicity may be the mechanism behind how synaptopathy actually is happening. But what's happening at the cellular level? Why do spiral ganglion neurons degenerate? Studies have shown that when you give a cell excess glutamate, it leads to a sharp decline in ATP. This causes a cascade of events that activates AMP kinase, and this is thought to mediate different reactions that cause ultimately the cell to undergo apoptosis. And it's interesting in the hair cell itself, why hair cells degenerate. It's thought to also be mediated by AMP kinase, and this leads to a different cascade of events that leads to hair cell death. So what are the functional effects of synaptopathy? What happens when you get synaptopathy? So the first thing that gets affected is intensity coding, and this is the ability of an auditory nerve fiber to encode sound intensity. And studies have shown that the ribbon survival percentage is directly correlated to the wave 1 amplitude. So what this means is the more survival you have of the ribbon of synapses, the larger your wave amplitude in an ABR. So the effect that you get when you have synaptopathy is you get a reduced amplitude of the brainstem response. And what this ends up manifesting clinically is that you end up having difficulty with sound perception. The second functional effect you get with synaptopathy is problems with temporal coding. And this is defined as the ability of a nerve fiber to follow quick changes in the acoustic signal. And in this study, not published by me, unfortunately, they measured nerves. They recorded from individual nerves of control animals in a guinea pig, and animals exposed to 1 day post noise, 1 week, and 1 month post noise. And what they found was that the latency of the response in these nerves was bigger compared to the control. And so the effect of temporal coding defects causes is that you have signal latency that's increased. And the clinical effect of this is that you start having difficulty with sound localization. And the last functional effect is preferential loss of auditory nerve fibers in the basal cochlea. So again, in this study, they looked at synapses, and they looked at mice that were exposed to noise, 0, 24, 1 week, 0 and 24 hours, and then 1 week and 2 weeks time points, that you had selective loss in the basal part of the basilar membrane of these auditory nerves. And then you also, this study showed in other parts that preferential loss of low SR nerve fibers happen as well. The clinical effect of this is that you get difficulty with discrimination and especially hearing and noise. And I want to bring up this slide from earlier on to demonstrate an interesting phenomenon here. And it's that when they actually measured the number of nerve fibers that degenerated, they calculated it to be about 40% from control. And like I mentioned before, 40% of all your nerve fibers are made up of low SR fibers. So this gave some more, this gave more of a clue that it's actually the low SR fibers that are being preferentially affected. But there's been another study that really shows and drives this point home that it's really the low SR fibers that are lost. And in this study, they measured from 300 different auditory nerves, and they wanted to understand what happens when you expose them to noise, which types of nerves go away. And what this is showing, these are tuning curves of the exposed guinea pigs and non-exposed guinea pigs, and they're all relatively the same. And these are showing you the thresholds, audiometric thresholds. So what this is showing is that when you expose animals to noise, the cochlear mechanics, which is the tuning, and the inner hair cell function, which is shown with the audiometric thresholds, are unaffected. But when you look at these individual nerves here, each of these represents one nerve they measured from. The low SR fibers are represented in this red triangle here. In the exposed, at the noise band, you actually get selective loss of these low SR nerve fibers. So this really shows that it is really the low SRs that are preferentially lost. So how does hidden hearing loss manifest clinically? So this is an audiogram that was kindly given to me by Dr. Fitzgerald. This is a patient he saw in clinic who's a 41-year-old lady who experienced tinnitus and difficulty hearing and noise after attending a concert without ear protection. And this is a audiogram that's very classic for what you see in hidden hearing loss. Your thresholds are preserved, like and this is a very normal audiogram. You have 100% word recognition in quiet, but when you look at the quicksyn, you actually see there's an abnormality there. And quicksyn, for those of you who aren't familiar, this is defined as the dB at which a person is able to identify at least 50% of keywords and sentences in the setting of background noise. So 2 dB is considered to be normal, but this person is having some difficulty with signal to noise ratio. They're not able to identify the signal in the background of noise. And this is considered mild signal to noise ratio loss. So this really drives home this point further. This is a figure, again, from Dr. Fitzgerald. This is showing you pure tone averages and thresholds on the y-axis as a function of quicksyn. And this is the figure that's still currently under review. But when you look at the people located here, they have normal thresholds. And they also have normal quicksyns. Like I mentioned before, 2 dB is considered to be normal. But you also have individuals in this range here where they have normal audiometric thresholds, but they have some moderate difficulty with signal to noise. So while this doesn't necessarily prove that this is hidden hearing loss and synaptopathy, because certainly these patients can be having difficulties with more central issues. They can be having more central issues. But it really shows you that the current audiogram and measuring your hearing and noise, in quiet may not be adequate enough. So I'll mention briefly some of the theories behind how synaptopathy is related to tinnitus. These authors tried to understand how synaptopathy could potentially mechanistically generate the tinnitus sensation. And this schematic here is showing you a drawing of an inner hair cell and its auditory nerve fibers. When we measure an ABR, the wave 1 is measuring the cochlear nerve, and the wave 5 is measuring the inferior colliculus and lateral lemniscus pathway. When you look at what these authors did was they got 15 healthy volunteers without tinnitus, and they measured their ABRs, and they found that their wave 1 ABR as expected was normal, and their wave 5 ABR also as expected was normal. But then they had 18 people with tinnitus, and they measured their ABR, and their wave 1 was expectedly low, as we had seen before. But interestingly, their wave 5 waveform was increased. So they realized that there must have been some response gain that happened in the system for this to go from a diminished response to an increased response. So they hypothesized that a decreased afferent signal must have led to some increased central compensation to eventually lead to tinnitus. And in their computational model, they actually found that, you have to have approximately, once you reach 60% auditory nerve denervation, you start gaining an increase of 1.3 of the signal that you had at wave 1. So in this study, it's really interesting to see these results because it's showing you a possible mechanism by which a tinnitus can be generated, and it may be related to the way we lose our synapses and our nerve fibers. So once you lose your nerve fibers, is that irreversible? Is that not able to regenerate? So this study found some interesting results in the guinea pig. And what they did was they wanted to see if there's any kind of spontaneous regeneration and plasticity within the mechanisms that can repair synapses. And what they're showing here is in the control, you have, again, a normal level of synapses. 1 day post-noise, you get a dramatic reduction in synapses. Then 1 week and 1 month post-noise, you actually start seeing a regeneration of these dots here. And this is showing you that this is a marker. This is the ribbon marker, and this is the postsynaptic density protein marker. And what they're showing is that you get a dramatic decrease initially 1 day post-noise, but you seem to recover these synapses after a month. And what's peculiar is that I want to take you back to this slide that I showed before. And if you remember, I talked about how you can have these long-term effects even a year after the even a year post-exposure. So why is this different? These authors actually replicated this exact same this experiment in mice. And remember, this was a study done in mice, and they found no regeneration in mice. So it's indicating that there are species-specific differences in the way that we can regenerate the synapses. There are some species that seem to be able to do that, while others not. And there are some promising therapies that could one day potentially go and help people with synaptopathy. But some studies in guinea pigs have shown that if you infuse neurotrophins in the cochlea, you can actually regenerate these synapses that are shown here. This is the neurotrophin group, and this is the control group after noise exposure. And this has also been done in mice. If you look at the normal synapses here, then if you expose the mice to loud noises, but give vehicle, you get reduction in synapses, but if you give neurotrophin 3, you can actually regenerate some of these synapses. So some promising therapeutics for the future. This is my last slide. I wanted to talk about occupational noise exposure because all of these studies really call into question what is an acceptable level of noise, especially in the work setting. So this is data from or this is the guidelines published by Occupational Safety and Health Administration. And what they define as permissible noise exposures is 90 dBs for 8 hours and 100 dBs for 2 hours. And we talked about that a lot of these animals have been exposed to these levels of noise, maybe even lower levels, and there was clearly evidence of synaptopathy at these sound levels. So to give you a sense of what 100 dB sounds like, 100 dB is like being next to a construction site. And 110 dBs would be, having loud music at a nightclub. For those of you who haven't been at a nightclub before but would like to know what that sounds like, you can always certainly ask our esteemed colleague Noel Ayub here. But before you do that, I recommend that you certainly have some hearing protection, because this can increase your risk of synaptopathy. So in summary, noise exposure causes loss of auditory nerve fibers. This causes increase in central gain, which can be a predisposition to tinnitus. Then you can also have some temporal and intensity coding difficulty or issues with that, which can cause poor sound localization, poor speech perception, and pitch perception. The inner ear synapse is, like I mentioned from these studies, the most vulnerable element in the cochlea, and it gets affected first in acoustic trauma. And it's preferential loss of low SR fibers that seem to be the pathophysiological mechanism behind this. So thank you all again for joining in on this presentation. I'd like to just thank my mentors. I'd like to thank Dr. Tony Ricci and Dr. Alan Chang for getting me excited about basic science research. I'd like to thank Dr. Fitzgerald for his help with putting this talk together. And I'd like to thank just the rest of the otology group for their encouragement and mentorship and inspiring me to pursue this field. So thanks again, and I'm open to any questions.
Well done, Johan. Very well done.
Thank you, Dr. Shirago. For some reason, I'm only able to hear you, Dr. Shirago. I'm not able to hear.
Excellent job, Johan. I hope you can hear me too.
Oh yeah, I can hear you, Dr.
I'll weigh in as well. I think we all muted our microphones, Johan, to eliminate.
Oh, gotcha. Okay, great.
No, that's great stuff there. Look, let me know when you're ready for questions.
Yeah, go for it.
Yeah, have you given, as you rightfully note, there's large parts of what we do clinically that we really can't assess it. Have you kind of given, what are your thoughts on then are things or tools that we could be looking at to try and assess synaptopathy more directly in humans, and then potentially what we might be able to do about it down the road?
So that's a great question. There's actually been studies that show that the wave 5 amplitude in the ABR is a very good sensitive indicator for synaptopathy. And I wasn't unfortunately able to show this today in this talk, but it may be that having an ABR done and measuring the wave 5 amplitude may be one way to measure synapses or measure the function of synapses in people in the future.
You mean wave 1, right? Not wave 5?
No, so actually they talk about how wave 5 is actually more sensitive than wave 1s. They say that wave 1s are actually not as reliable. It's actually interesting, because I thought it would be wave 1 that would be more reliable, but they say there's a study that shows it's actually with the wave 5 amplitude and the latency that's more sensitive to synaptopathy.
Yeah, historically, the challenge with using ABR wave amplitude just been the variability is so high between individuals that it's a distinct challenge. Kind of then if one person has, because depending upon where the electrodes are placed, right, how it's recorded, frankly, what the impedance is from the recording from time to time, and these things can all tremendously influence the amplitude of any waveform that you record clinically. So that's I agree that this is kind of, I think, probably the thing we can do clinically that's most likely to be sensitive to synaptopathy itself. But I think I speculate we're going to have to see some advances in in recording or getting a sense as to like what a true reduced amplitude is in this regard, because there's a lot of things that go into ABR amplitudes
Right.
with the system. So
Cool. I have a message from Dr. Chang. Is there evidence that synaptopathy can be reversible in humans? I didn't find any, I actually was curious about that. I actually couldn't find a paper with that. I think the problem is that it's very difficult to study in humans for obvious reasons. And there's not a good measure, like Dr. Fitzgerald just mentioned, of knowing, or first of all, measuring synaptopathy and knowing when there's a change, you know. So in animals, it's easy because you just sacrifice them and take a look at their ears, but certainly can't do that to humans. So.
Very nice, Johan. I have a question too. I wasn't aware of the guinea pig study. So how well did the histology recovery correlate with a recovery of the function by ABR? Did they look at that too?
I believe, I have to double check that. I believe they did do an ABR, and that, sorry, so they did do ABRs, but ABRs or any kind of thresholds, the problem is that it doesn't capture synaptopathy well, right? So even, I don't remember exactly what the audiometric outcome was here, but like I've shown before, even when you have threshold recovery, it doesn't actually mean that there isn't a change happening in the synapses. So that's the issue, I think.
Very nice.
Joanne, did you study anything about the effect of intratympanic steroids immediately after noise exposure?
That is a great question. I don't think there's any studies out there, at least to my understanding, that looks at whether steroid infusions can, you know, help or reduce the level of synaptopathy. But that's something definitely that I think should be looked at. All right. Well, I don't see any more questions. So thank you, everyone. I try to finish a little bit early so you can have the evening off. Thanks again, and see you guys soon again.
great work, Johan. Great stuff.
Good job, Johan.
Great job.
Well done, Johan. Really.
Thank you.